So I find that a lot of students have no trouble understanding the phases of cell division, but they don't understand sort of the organization of the chromosome. So I wanted to take some time to make a special video just about that. So first of all, let's remember our DNA. So our DNA is made of 2 strands. Building block is nucleotides. And so nucleotides have partners that they bind with. So for example, if we have G here, we have C there. If we have A here, we have T here. And so those are going to follow our base pairing rules. But we can't just stuff our DNA into our cell. So this is our 2-stranded, let's write it down here, 2-stranded double helix DNA. All right, so we can't just stuff it in a cell, we need to access it. So for example, we might have gene 1 over here and gene 2 over here, and we have to be able to get access to all of our genes in order to turn them off or on when we need to. So when we organize it and stuff it all in the nucleus, we have to have a lot of order, or we won't be able to get to the genes when we need them. And so one of the ways that the cell organizes the DNA is that it wraps it around some proteins, and these proteins are called histones. So a lot of times you'll see in pictures that there's 4 histones. Behind that there are another 4. So there's 8 histones in this complex. And then the DNA wraps itself around those histones. So I'm going to draw a dark line for my DNA wrapping around those 4 that you can see, 8 histones. And so my dark black line there is going to represent this double helix. So this is what we're seeing here, right? And so if you imagine, we're then eventually going to have these little pieces here, histones, that are going to kind of look like beads, and the DNA is wrapping around them. And so this is called chromatin. Chromatin is the DNA plus the proteins, and then it keeps wrapping on itself, right? So now you can think of this as a piece of rope, and it's going to wrap around itself as well, right? So we now have more wrapping and more wrapping, right? And so ultimately that's going to make a chromosome. And the way we will start talking about chromosomes, just to have a simple way to talk about them, the way I'm going to draw them is as just a line, right? And that line is going to have a structure at the center of it called the centromere. And that centromere can be in different places for different chromosomes, right? Those are still centromeres. Let me make sure here, right? Either way. And that is going to help us sort of separate our chromosomes later. So when I draw a chromosome like this, this is what I mean as one copy of a chromosome. So one of our phases that we've already talked about with cell division is we've talked about replicating or copying our chromosomes. And so what we would do when we actually want to replicate our DNA, and I'm going to move back up here for a minute. If I start with one chromosome like this, right? This is a single copy of a chromosome with a centromere. And if I want to copy it, I make a duplicate of it, but they're still attached to each other. So they often look like this. Still have 2 centromeres, but they're stuck together. So what I like to call this is a stick, and this one is an X. So sometimes I talk about sticks and X's. And so when you're thinking about some of the problems that I'm going to give you on during this chapter or on the test, you've got to ask yourself, depending on which time you are looking at these chromosomes, are they going to be a stick or an X? And so an X is a duplicated chromosome, and a stick is a single chromosome, right? And so a duplicated chromosome has identical, each side of the X. So if this is gene A here, then we have an identical copy of it there. And so we can say we have X on this side, and we have an identical copy of X on our other side. So the X, one side of the X is identical to the other. And so when we copy them during S phase of the cell cycle, that's what we want, because these two new copies are going to go into the cell that's dividing. So one cell will get one stick, and the other cell will get the other stick. So when we divide it, right? And so let's sort of look at what that would look like. So if we have our duplicated chromosome here, it has identical copies, A here and X here. During that process, we're going to pull this one and this one in two directions. So that means we'll end up with A and X over here, and then the same thing on this side, A and X. So this will be part of a new cell, and this will be part of a new cell. When you're doing problems on this, what you will find out is oftentimes, to set up the problem to understand it, one of the first things you have to do is determine what your chromosomes look like. Do they look like this, or do they look like this? And then after you figured that out, a lot of times it's just pretty easy. But I will say that this is complicated partly because of the terminology that's used, and we'll see when we get there. But what I would say first and foremost is that if you have a cell that has already gone through S phase, then you need to think of all your chromosomes as being duplicated chromosomes. And they won't go back to being single chromosomes until they get to the very end of the cell cycle and get separated into two new cells. I'll see you again and talk a little bit more about different phases and what's going to happen there.